Buonasera a tutti e benvenuti a Montecitorio nella sala stampa. Saluto il dottor Bertelli che ha voluto portare in questa sede, che a me piace definire la casa degli italiani, la presentazione di una nuova realtà di ricerca che si articola attraverso la collaborazione tra scienziati dislocati appunto tra l'Italia e gli Stati Uniti, dove io sono stata eletta e infatti rappresento gli italiani che vivono anche negli Stati Uniti, in Nord e Centro America e negli Stati Uniti. Pertanto sono davvero orgogliosa che si sia instaurata una collaborazione tra le due sponde dell'oceano, a partire da quelle che sono le risorse naturali tipiche del nostro contesto e che sono comprese in quella che è universalmente riconosciuta come dieta mediterranea. Tale dieta non è solo alimentazione ma anche modo di vita, tanto che l'UNESCO, nell'iscrivere la dieta mediterranea nell'elenco del patrimonio immateriale dell'umanità, affermava che è molto più che un semplice elenco di alimenti. Infatti promuove l'interazione sociale, poiché il pasto in comune è alla base dei costumi sociali e delle festività condivise da una data comunità e ha dato luogo a un notevole corpus di conoscenze, canzoni, massime, racconti e leggende. La dieta si fonda nel rispetto per il territorio e la biodiversità e garantisce la conservazione e lo sviluppo delle attività tradizionali e dei mestieri. Quindi, per me che sono impegnata nel preservare e promuovere il nostro cultural heritage negli Stati Uniti, questa conferenza ha un valore molto importante e sono qui per augurarvi buon lavoro a tutti voi, a tutti i ricercatori impegnati a dare appunto riconoscimento scientifico a quegli elementi naturali che caratterizzano la dieta mediterranea. Ringrazio pertanto tutti coloro che sono impegnati in questo lavoro e in modo particolare i ricercatori in prima linea sul territorio americano. Dal lavoro operato trarrà sicuramente giovamento il nostro sistema paese in termini di promozione della nostra cultura e delle nostre tradizioni. Quindi cedo la parola al moderatore, che ringrazio, il dottor Ludovico Seppilli. Grazie.
Thank you. I speak English in order to our guests from Atlanta. Thank you. Thank you, Honorable Nissoli, a brilliant member of our parliament. Thank you also for the opportunity to have this conference here in the Chamber of Deputies. And thank you, Mr. Bertelli, for this initiative, an important opportunity to highlight the value of relations between Italy and United States and between Georgia and the United States. So I'm here to make sure everything works with maximum respect of the time we have. So please let me remind all the speakers to keep their speech within 3 minutes. Honorable Nissoli, 3 minutes, very good. So everyone please take note and do the same. Thank you, Mr. Bertelli. I leave the floor to you for an introduction. Okay.
Or to open my presentation.
La presentazione è quella che avete voi. Sì.
First slide, please. I need the slide.
Sì.
No,
Questa qui no, è quella che è arrivata via mail a voi.
we have in our.
Qui al volo.
In a typical Italian organization, I present the science behind the natural molecule and Mediterranean diet. Please, the first slide. Thank you. Good evening, everyone. Today I'm introducing the new spin-off MagisNet based in Atlanta Tech Park. Next slide, please. Our mission is to carry out research on molecules naturally contained in a typical plant of the Mediterranean diet. Our scientific approach is to apply omics in the study of this natural molecule. Our company has the FDA certification and registration and also is a member of CRN. Next slide. We formulated two dietary supplements, Garlive oral spray with polyphenol from olive tree and Garlive recovery in tablets with polyphenols and vitamins. This dietary supplement support healthy immune system. MagisNet business plan is focused first on studying natural molecule in the context of eating disorder or lipidema. Second, in the promoting Mediterranean diet by organizing meeting and course about it. And last, marketing of dietary supplement. Next slide. With this last slide, I want to introduce and thanks the speakers, all professors from Italy and USA who are attending our press conference. Thank you.
Thank you very much, Mr. Bertelli. So now I'm going to give the floor to Professor Connelly. Hope, Professor Connelly, you can lead us, you can hear us loud and clear. Sì, proviamo con questa.
Yes, I can hear you. Okay, look,
Professor Connelly, can you hear us?
I sure can. Let's see. Can you hear me? Let's see. All right. Hello. Can you hear me now? Oops. I'm muted. See, I think, can everybody hear me now?
Yes.
Okay, very good. Well, it's an honor to be here today. And Dr. Bertelli has really been working very hard on something very special. And it's been an honor to work with him on his projects. And I look forward to him and his team improving the lives of people through the Mediterranean diet and the Mediterranean nutritional supplements that people will need to help them change their lives. And I appreciate it, and I'll keep my remarks short in respect of time.
Thank you very much. Thank you, Professor Connelly. So now we have to move to commercial relations between United States and Italy, particularly Georgia and Italy. I really would like to thank our honorary consul in Atlanta, Filiberto Calascibetta, and our Italy-United States Chamber of Commerce president in Georgia, Mr. Eugenio Fumo, for joining us directly from Atlanta. So I give the floor to our honorary consul, Mr. Filiberto Calascibetta. Hope, okay, yes, I see you. So I think you can hear us. Okay, okay, yes, we see you. Ma non sentiamo.
Sì, mi sentite?
Sì, un po' basso.
Ah,
Ecco, adesso bene, adesso bene.
bene, bene. Grazie per l'invito, è un onore essere qua oggi, questo intervento e questo scambio di importanti informazioni sull'iniziativa imprenditoriale, ma è anche nella mia veste ruolo e responsabilità di console onorario, che rappresento le istituzioni, e il console onorario per lo Stato della Georgia qua nell'area metropolitana di Atlanta. Volevo cogliere per condividere con i presenti che l'iniziativa imprenditoriale, l'importante iniziativa imprenditoriale e scientifica appunto rappresentata è a mio avviso, ma non solo mio, di un po' l'espressione di quello che è lo sviluppo e la strada di crescita e di rafforzamento della realtà imprenditoriale e della comunità d'affari e comunità scientifica italo-americana nello stato della Georgia. Come rappresentante della comunità italiana qua in Atlanta, in Georgia, posso senz'altro esprimere questa evoluzione positiva negli ultimi 10-20 anni in realtà della realtà in cui viviamo, e la Georgia è un'area geografica che offre tante opportunità dal punto di vista non solo imprenditoriale e scientifico, ma anche culturale. Abbiamo appunto una presenza italiana parecchio in crescita, e volevo ringraziare appunto per l'invito, anche appunto nel nome del Consolato Generale di Miami, che ha supportato, il Console Generale Cristiano Musillo, che ha supportato davvero con tanta energia e determinazione la realizzazione di questo rafforzamento della presenza italiana nello Stato della Georgia, anche con la creazione della Camera di Commercio e del chapter della Camera di Commercio italo-americana che è presente qua in Georgia, che appunto supporta la realtà di imprenditoria e di investitori italiani nell'area di Atlanta, appunto nell'intero stato della Georgia, e poi è posizionata appunto nella posizione strategica nel sud-est, nell'intero sud-est. Grazie per l'attenzione. Io passerei la parola con un commento un po' più dettagliato al Presidente della Camera di Commercio, Eugenio Fumo.
Grazie e buongiorno, spero che l'audio sia ok.
Sentiamo bene, sì, sì, yes.
Benissimo, allora continuo in italiano anch'io, a meno che non sia idoneo farlo in inglese, ma insomma cioè il Parlamento italiano mi sembra giusto portare avanti la conversazione in italiano. Allora, il mio nome è Eugenio Fumo e rappresento la business community italiana nello stato della Georgia. Noi siamo una piccola Camera di Commercio fatta di membri volontari che hanno pensato di riunirsi per dare diciamo un'impronta, un'immagine a quella che è una comunità e un tessuto industriale italiano che si è sviluppato parecchio nell'ultimo ventennio. La Georgia, come detto, il console onorario è uno stato estremamente importante nella regione sud-est ed è uno stato che è cresciuto incredibilmente a discapito di altri stati che invece hanno visto una migrazione interna. Credo di poter dire che la Georgia sia il secondo stato negli ultimi 10 anni per crescita di popolazione, venendo solo dopo il Texas. E quindi le aziende italiane hanno cercato di adattarsi a questo trend, hanno capito che l'America era fatta di più città che delle solite 5 o 6 note a tutti, e che in effetti se volevano intraprendere un'attività produttiva, esistevano condizioni molto più favorevoli a sud, e nello specifico nella Georgia, rispetto a tanti altri stati. E quindi con un po' di senso di avventura si sono instaurati qui e sono cresciuti negli anni, giovando di tante cose. Da un punto di vista logistico la Georgia è ben posizionata con circa 3 porti abbastanza vicini e quindi che ne favoriscono lo scambio commerciale di merci e così via. E poi chiaramente con l'esploit anche dell'aeroporto di Atlanta, che pre-Covid era il numero 1 nel mondo a livello di passeggeri, con una compagnia aerea che sicuramente era tra la prima e la seconda nel mondo a livello di numero di voli, e quindi favoriva molto, molto facilmente anche il trasferimento sul territorio nazionale e internazionale di quei business people che vogliono chiaramente, che devono viaggiare per lavoro. Quindi è sicuramente stato una cosa favorevole per l'Italia, credo, scoprire la Georgia, ma anche per la Georgia, che ha inseguito tanti investitori italiani in un modo veramente proattivo. Quindi la Georgia è molto pro-business a livello di volontà di attirare investitori esteri, e sicuramente rimane un posto molto, molto più economico rispetto a tanti altri stati, una tassazione molto più bassa che ne favorisce quindi lo sviluppo, e sicuramente anche un supporto locale dalle istituzioni alle aziende molto sentito. Non è inusuale che in queste piccole città della Georgia il sindaco non solo vada a trovare l'imprenditore qui in Georgia, ma che prenda anche un volo per andarlo a trovare in Italia. Quindi ho sentito tante belle testimonianze da parte di imprenditori italiani che sono rimasti molto ben impressionati dalla volontà di instaurare un rapporto anche con proprio la parte diciamo statale della Georgia. Quindi credo questo in un breve intervento possa dare una panoramica. Sicuramente anch'io ringrazio l'operato responsabile Musillo, che ha facilitato l'istituzione di questa Camera di Commercio, che fa comunque parte anche in questo caso a quella di Miami, e che quindi spero possa mettere una piccola bandiera italiana anche per la business community italiana in Georgia. Grazie.
Grazie. Thank you very much, Mr. Fumo. So now we have the opportunity to listen and focus about lipodema between genetics and nutrition from Professor Karen Herbst. Okay, we can see you. Hope you can hear us. And I give you the floor, please.
Thank you. Can you see my screen?
Yes.
Thank you.
So your presentation is loading, I think.
Okay. Okay, great. So as Dr. Bertelli mentioned, I'm going to talk briefly about...
Okay, now it works. Here, okay, please.
Okay. So as Dr. Bertelli, well, first of all, thank you so much for this invitation. It's really an honor to be here. And as Dr. Bertelli mentioned, we're very interested in using the Mediterranean diet and the supplements which he showed for the treatment of lipedema, because there are very few treatments for lipedema except for surgery. And as you can see in the picture, this is a woman with lipedema. She's got a disproportionate increase in tissue on her legs. This tissue is very inflamed and it is very fibrotic, making it difficult to lose by diet, exercise, and even bariatric surgery. There is edema in the tissue. It's painful. It bruises easy. It affects millions of women. So it's proposed that the prevalence is 5 to 10% of females. And it is inherited in 60%, probably even more than that. And these are a list of genes of interest. They've not been proved as a cause of lipedema except for one shown here, and mutations in this gene were found by the MAGI group. And when this gene is mutated, progesterone levels increase, and that is known to increase adipogenesis. And this gene was discovered by the MAGI group. And lipedema is an inflammatory disease. This is a diagram just of a general marker showing that overall there are more inflammatory cells in lipedema leg tissue than in controls. And there may even be some inflamed tissue in the abdomen as well, but not quite as much, which did not reach significance. And there are two different groups that have shown that there are increased macrophages in the tissue. On the right side, you can see that there are increased macrophages, which are a type of immune cell in lipedema tissue. And it doesn't matter whether they have obesity or they don't have obesity. So this is not an obesity-driven inflammation. It's a lipedema tissue-driven inflammation. And the same thing on the right side, or sorry, left side of the photo, you can see that in general, there are more white blood cells on the very left, and then the second to left panel shows an increase again in macrophages in lipedema tissue. Just to drive home the point that lipedema is inflammatory, there's a marker called platelet factor 4 that was shown to be higher in patients with lipedema compared to controls. And also this depend on obesity, it was independent of obesity. There is one on the Mediterranean diet in women with lipedema, and they did lose weight, as did the control group. But unfortunately, while the control group lost % of fat tissue all over the body, women with lipedema did not. So we need additional treatments, including anti-inflammatory supplements, to help reduce the inflammation and allow it to be lost by diet. Also, there's no significant reduction in pain. So women with lipedema have inflammation causing their fat tissue to be difficult to lose by diet or exercise. Natural molecules with anti-inflammatory potential based on the Mediterranean diet are needed to reduce inflammation in lipedema and lymphedema tissue. And we think a balanced, healthy, anti-inflammatory diet, especially a Mediterranean-based diet, is important to prevent and treat lipedema and lymphedema. Thank you.
Thank you very much, Professor Herbst. So now we have nutrition, inherited retinal degeneration, a new therapeutic paradigm. So Professor Falsini, you can join us. I have your presentation just right here.
So, thank you. First of all, I would like to thank the organizing committee, Professor Bertelli, for the kind invitation to participate in this important meeting. And I just would like to spend a few words about our focus of research. She's now on nutrition in inherited retinal degeneration, and the idea to develop a potential new therapeutic paradigm. Can we go ahead with slides? So the disease is characterized by a progressive loss of retinal photoreceptors. So the cells that are extremely neuronal cells that are extremely important for vision. So we have a large sample of rare patients. So the disease is rare, but it's not so rare. So we have a widespread presence of retinal degeneration in both Europe and United States. And we know that in the typical time course of the disease, photoreceptor degeneration affects first rods and then cones. And then cones start to degenerate and dying for apoptosis after more than 75% of rods are lost. we focused our attention on the nutritional aspects of the disease in a large cohort of retinitis pigmentosa patients followed clinically at Policlinico Gemelli in Rome, with a strong collaboration also with other centers, including the Children's Hospital of Bambin Gesù in Rome and Tor Vergata University, headed by Professor Cusumano. Ahead, can we go ahead quickly? So there are many hypotheses to explain the death of photoreceptors. One important is related to the metabolic dysregulation of these cells. Can we go ahead again? And what I wanted to say, by the way, is that photoreceptors do not die immediately, but they have a long period where they lose the ability to catch the light and to help in vision, but they are still alive. So we may also rescue these cells with appropriate interventions. Can we go ahead? And it has been demonstrated in several animal models studied in the United States and now also in Italy that some modification of the metabolic pathways within the photoreceptors, including the adenosine monophosphate kinase pathway, which is extremely important for the metabolic regulation of photoreceptors, it might be important to rescue the cells, the retinal cells, and to prolong the survival of these cells. This means that if we can apply this nutritional paradigm in humans, mimicking the effect of activation of the adenosine monophosphate kinase pathway, we should be able to prolong the survival of cone photoreceptors in patients with inherited retinal degeneration. Of course, not in all patients, because there could be a genetic specificity for this correction. But we started a pilot study. Can we go ahead? A second, another slide. A pilot study measuring cone electrophysiological cone function in a large cohort of patients with retinal degeneration and looking at their nutritional status. And to be short, because the study is still underway, we found that the best cone function adjusting for the other clinical variables can be observed in patients with what we call dietary restriction or caloric restriction that mimics the activation of the MP kinase pathway, like, for example, some fundamental constituents of Mediterranean diet, like, for example, antioxidants, because we know that the diet is rich in antioxidants, and they can activate in the same manner this metabolic pathway. So our paradigm that we are working on is a paradigm based on modifying the metabolic pathway of cone photoreceptors by acting on the nutritional status of the patient. This is our project, and we hope to go ahead and to bring good results in terms also of their impact in the business of nutrition. Thank you.
Thank you, Professor, for your focus on your research. And I ask Professor Cusumano to join us here and continue about nutrition and retinal degeneration.
Just one presentation.
Yeah, okay. Just if you want to add something, or if you want that.
No, I can just tell you that we are moving in a very fascinating and innovative thing. Basically, we are working since many years in trying to rescue photoreceptors with many different compounds. We are specialized in protection for ultraviolets. We know that they are very sensitive. But I think that now with the perspective that has been presented today, we are moving in an unusual, consistent direction that can also be joined by a large group of international potential patients, and especially in countries where maybe there are an overexposition to phototoxic damage of photoreceptors, and where maybe there are not so large possibilities of using different protectors. The new dietary system and approach will be the right one for the moment, and has been underevaluated, but will take the good room for the next years. And we hope that we can continue our studies together. Thank you.
Thank you. Thank you very much. And so now we have the honor to have with us Professor John Paul San Giovanni from the University of Arizona. Professor, hear us? Okay, we can see you.
Yes, thank you.
Thank you. So please.
So if I'm attempting to share my my screen, and this will just take a second. And
no problem. Okay, now we see your screen loading, and
okay, just to make sure.
I think in a couple of seconds we can see your screen.
Okay, I'm hoping to show my slides now, and what you should see. Do you see my PowerPoint?
Not yet, but
okay. I will wait.
Okay, now we see it.
Okay, so I'm actually trying to display it myself so I can see it, but for some reason, okay, so I'm here. Thank you very much. And I will start my presentation. So first of all, while I'm just loading the presentation, I'd like to thank...
Okay, perfect. Now we see it very clearly. Thank you.
Okay. Fantastic. Thank you. So it's my great honor to be here, Deputy Nissoli, Dr. Bertelli, thank you so much. You know, you talk about the science behind the natural molecules, and this is something of great personal relevance to me because it has been a way of life throughout my life. My family has its origins in the Mediterranean basin, and despite a family history of cardiovascular disease and cancer, very bad cancer, we all seem to live into our late 80s. And so I'd like to just say that I really think that this approach is germane. You know, in the context of molecular profiling technologies that are now becoming accessible, and the properties of these technologies can tell us so much about the underlying biology and causative biology. I'm very excited, and I just would like to say that I'm committed to this, and I think this is something that has tremendous promise. The other quick comment that I'd like to make is that you know, Atlanta as a center of excellence in this unit is something that I completely support as well. My son ended up attending Emory University. He only applied to one school. He could have gone many places, but he chose to go there because of the quality of the research, both human and basic. So with that in mind, I'd like to speak about, you know, my approach to linking foods of the Mediterranean basin and the Mediterranean basin and their nutrients to these physiologically responsive targets in the body. What I thought I'd do is basically start with the message. We're applying what's known as a deep learning approach to develop what are known as quantitative structural activity models, which basically allow us to leverage data on what we know as the chemistry in the foods, the three-dimensional structure of the chemicals in the foods, to places in the body, receptors that actually are key in underlying some physiologic processes that keep our cells, that rescue our cells, that keep our cells working as they should, and also act in ways to affect metabolism. So the bottom line and the take-home message is that, through this approach, we were able to identify a compound, a promising compound that's present. It's known as ferulic acid, and the name is based upon it's a phenolic compound, and it's based upon the genus Ferula, which is coming from the giant fennel, Finocchio. And so this is something that was and is a big part of my diet. I know it's on your table. Ferulic acid is also found in oregano. It's found in appreciable quantities in olives. So it's the spices, it's the foods, and also the chechi. We can find it there. It's very interesting that if we leverage this information about the structural chemistry, we have identified a key protein that's involved in angiogenesis. And so the endpoint, or my major focus of research, is on macular degeneration, which is a blinding eye disease that affects millions of people. And there are a lot of the healthcare expenditure it's millions and millions of US dollars and euros. There are millions of people that lose their sight to this disease. So what was very interesting is that through our structure activity analysis and through our machine learning, we identified this target chemical relationship, the chemical being the ferulic acid, which is present in these foods of the Mediterranean basin, and the target being this thing called a gelatinase, which basically clears the space for new vessels to grow. And so that's the message. Now, I just wanted to tell you about the rigor of the method and how we do this. So as I said, we're leveraging data from these chemical databases. And so there is a whole process that is used to actually identify and characterize the three-dimensional structure of the chemicals in foods. And as it turns out, this slide was based upon something that I saw in a presentation from a colleague in Atlanta from Emory University, but it is basically a very rigorous process that allows us to make assumptions about the structure, the variation in that structure, and the complexity of factors surrounding that. And so I'm going to try to advance, and it's not advancing at the moment. I'm just trying to see, pardon me. So, okay, fantastic. Okay, so I'm trying my best to keep this short, but I just like to say that my ancestors are from the Mezzogiorno. So, okay, so basically, just to give you some numbers here, we have data on these on the structural chemistry on about 800 foods, and these encompass over about 71,000 compounds. There are approximately 4,000 compounds for which we have quantitative estimates. So it's one thing to say that a compound is there, but then we have more precise information on how much is there. I talked a little bit about how we looked at the target relationships. And so, but I just wanted to point out that when we looked for the connection or the overlap between the chemical and the receptor, what we were using was retinal tissue from donors who had this macular degeneration, this disease, this macular degeneration. So we were actually looking in the cells, we were looking at what are known as the transcripts in the cells that actually manifest the disease itself. And we had information from patients. We also have information on DNA variation that comes from blood. So we were very interested to see if the MMP, this compound that affects the pathology, if we had variation that was specific as a profile that defined people with macular degeneration. And as it turns out, we had to answer questions on plausibility and the pathophysiology. So as it turns out, there's more of this MMP in people with disease. And it's, again, I said it's important for clearing the way for these vessels. It's increased about 1.5-fold. And we also have 2 variations in DNA that affect the coding or the code to make the protein. So not all DNA is used to make protein, but we have two instances and they're significant on what's known as a genome-wide level. For people in science at 10 to the minus 8, 10 to the minus 7. And so we now understand that this is a plausible opportunity. So now we had to also look at validation. So there were studies that looked at ferulic acid. So if there's more ferulic acid in people with macular degeneration, if we have what are known as DNA sequence variants that affect the coding, the structure and the volume of the protein or the abundance of the protein, we need to see what this constituent in foods may do. And there was a study that was not done in retinal cells that actually showed that ferulic acid binds to MMP and it basically reduces the activity of the protein. So that's what we want. Okay, so the next step is what I'm doing now in my lab, which is another validation in which we're taking human cells, and I'm trying to advance the slide again, forgive me, but we're taking human cells that were donors, again, from a study that I conducted. I was the scientific lead at a study at the National Institutes of Health. And what we're doing is we're converting these stem cells into a retinal system on a chip. And unfortunately, this is not advancing, but we have a paradigm that will allow us to take cells from people with macular degeneration, make these vascular networks as they would exist in the living human, and then apply different concentrations of ferulic acid. And so, unfortunately, again, I'm very sorry, but this, oh, here we go. So basically, this is the paradigm. We're using something called the Mimeta system. And so we hope to get that in place this summer. We're using some very advanced imaging technology that will allow us to image the cells over a 5-day period. So the idea is that we've gone from this compound that is resident in typical Mediterranean foods to a target, and it's a physiologically relevant target for macular degeneration. And then again, the validation that this compound ferulic acid actually blocks a pathologic compound. And at the point we are now, we're just going into the human systems. And with that, I'd like to just thank my collaborators. I know I'm out of time, but it's been a great pleasure to have this opportunity. Thank you so much.
Thank you, Professor. I gave you some minutes more, but you asked me for it, so I have to tell it. Okay, now we can go on, and I ask Professor Peng Jin about natural molecules as dietary support. Professor, can you hear us? I see you. Okay.
Can you hear me?
Yes, I can hear you. So I give you the floor, and please.
Thank you. First of all, I want to thank Dr. Bertelli for providing me this opportunity. I'm really honored to be here. And so for my research, actually, I'm based in Atlanta, and I'm a professor and a chair at the Department of Human Genetics at Emory University in Atlanta. So my lab really focuses on the Fragile-X-related disorders. In particularly, we have long interest in neurodegenerative disorder called Fragile-X-associated tremor ataxia syndrome. So this, which is a late onset neurodegenerative disorder with, developed within the pre-mutation carriers of the Fragile-X genes, and which has a long CGG repeat. And so those patients were typically develop the gait ataxia, intention tremor, and the Parkinson phenotypes. So we have studied this disorder for the past 20 years, and unfortunately, there's still really no treatment for this disorder. And so my lab has really developed multiple different model systems to study the molecular pathogenesis of factors, including fruit fly model, mouse model and the human iPSL-derived models. And so several years ago, actually, we did a small molecule screening, and we have identified some kind of natural molecules actually that could potentially modulate the neuronal toxicity associated with the Fragile X with the factors patients. So that's actually led us to really think about more broadly whether natural molecules could serve as a therapeutic or treatment for factors. So I think it was very excited. I was very excited to meet Dr. Bertelli and really discuss and establish our collaboration to really test polyphenols, the potential role, therapeutic roles in treating in treating factors. So now, since I'm based in Atlanta, so we plan to utilize the small molecules developed by Dr. Bertelli to really test in multiple model systems that we have established in the lab, including fly, mouse, and human iPS cells. So I think this could really potentially lead very exciting discoveries and therapeutic development for factors. So I really look forward to the future collaborations with the company and Dr. Bertelli. Thank you.
Thank you, Professor Jean. And so our last speech, but not last but not least, exactly. So Professor Pietro Curazzi from Policlinico Gemelli, and so
a good friend of Professor Jean as well. So I also worked for many years, almost 30 years, on Fragile X syndrome. And maybe there could be still some time to show the slides. Yeah. Anyhow, it's important to remember. Yeah, you can go ahead, please, with the next slide. This is just to show on the X chromosome how many genes are already located that cause intellectual disability. And Fragile X is just caused by the first of these genes, FMR1, which is indicated by the big red arrow at the end of the chromosome. And this was cloned or identified in 1991, so 31 years ago. This was the first gene identified on the X that caused intellectual disability. Now we have more than 130. But this is still a very relatively common rare disease. So can I have the next slide, please? Yes. So it's important to know that this is a repeat expansion disorder. So this means that there is a repeated sequence, namely a cytosine followed by 2 guanines, CGG, that are normally repeated around 30 times in our DNA. And it's in which region of this gene. So basically the switch that turns it on or off. When this sequence is growing larger, it can reach over 200 repeats. In that case, the switch is off. The gene is shut down and you're not having the protein, and this causes intellectual disability. But we're not talking about these individuals here. We're talking about those individuals who have around 100 repeats. These are so-called pre-mutations. That's the third panel from top or the second from bottom. Can I have the next slide, please? In that case, now it's the panel on top. When you have around 100 repeats, the gene is still on. The protein is there. You don't have intellectual disability, but you are stressing your neurons. And your neurons eventually start dying out. And this happens with age. So this will cause eventually Parkinsonism or tremors. And it will also cause a cerebellar disease with ataxia. That means unbalance and gait as if you were drunk. And in order to prevent this neurodegeneration of this condition, which is called FAXTAS or Fragile X-related tremor ataxia syndrome, a very horrible name, we thought that antioxidants might be very useful. So can I have the next slide, please? Yeah, so these are just a couple of papers that illustrated one of those, it's ours, but they are just, I don't assume that you read them, but believe me, they are telling that mitochondria, which are basically the metabolic engines in our cells, the factory of energy, are working not 100%, and they are actually accumulating oxidative radicals that will damage eventually relevant cells, that is neurons. So the next slide. This is a nice study done by a group in UC Davis, a colleague of mine, Flora Tassone, that originated in our lab at the Gemelli Hospital, and now she moved already more than 20 years ago to the US. And they really looked also at the metabolic profiles of these individuals. And I know that Dr. Bertelli is a fan of metabolic profiles. He eventually grew very fond of studying the metabolomics and not only the genomics, from which he started as a colleague and as a medical geneticist. And in the next slide, please, that's my last, I think. Although you can't read it, believe me, we are talking about hydroxytyrosol, which is one of the many phenolic components of the wastewaters from olive oil extraction. So basically, is a waste product that you would throw away after the processing of the good olive oil. And it has a very good effect as antioxidant. And the article on the right is actually relating on a study on patients with heart disease. And it's also good for prevention of heart disease. It's actually a group from Spain. There you go. They also have, they have a lot of olive oil, although we think that ours is best, but it's still the Mediterranean. And in the last slide, very last, thank you, the last one. Yeah, that's important because I think from a business perspective, we are also valorizing, putting actually value into some waste product. It's really in the title, wastewater from the olive mill. They, instead of throwing it away, they could become a source of dietary supplements. And our intention in collaboration with Maggi is not, and Dr. Bertelli and Professor Penjing in Atlanta, is to try these compounds and these fractions, phenolic fractions, on all the disease models for FACSTAS that are available in his lab. And so with this, I thank you very much for the invitation and for the opportunity to talk in this special place. Grazie.
Thank you, Professor Chirazzi. I ask Dr. Bertelli to have another comment for us.
Yes, I'm particularly grateful to Onorevole Nissoli and the Chamber of Deputies, Italian Chamber of Deputies, for host this event. And also want to thank all the speakers, professors from Italy and USA. And in particular, I want to thank Professor Stephen Connelly for his fundamental effort and help in develop of MagisNet project. And also want to particular thanks Professor Penjin for open us a possibility for a future collaboration in Atlanta. And also want to thank all the people who participate in this event and demonstrate how is important the nutrition in our life.
Thank you, Dr. Bertelli. Everybody know the quote from Feuerbach, "We are what we eat." And so the Mediterranean diet was born in Italy, but is particularly appreciated and studied in the United States. So I think that this massive work MagisNet is doing and will continue to do in the next months and years, opening its activities in the Atlanta Tech Park in Georgia. I think it's an opportunity for our commercial, economic, institutional relations, but also for research. And so I would like to thank all the professors and researchers involved on these fields and during the conference today. So thank you very much, Mr. Bertelli. Thank you, everybody. Huge thanks also to those who are following us from the Tech Park, from Atlanta, directly from Atlanta, and hope to attend an event like this in Georgia and Atlanta as soon as possible. Thank you very much, and thank you, Honorable Nissoli, for the opportunity to be here today.